I'm talking today about some work that I've been doing with Convergence and Lisa Gerstein, and just looking at why convergence evolves. So a lot of us are probably familiar with things like MC1R, which is a gene that drives color pattern polymorphism in organisms as widespread as mice, lizards, snakes, fish, even woolly mammoths seems to be. And it's seeing this convergent genomic evolution over and over again being driven by MC1R is kind of fascinating. It implies all sorts of things about constraints. And what I want to think about with this talk is just why MC1R? Why do we see a gene like this over and over again? Or in other cases, other examples of convergence, why other genes? Some nice work from Hopi Hoekstra kind of summarized a whole bunch of different genes in mice that can drive color polymorphism. So we know there's many, many different ways you can change color polymorphism in mice, but over and over again, they see MC1R coming up as one of the genes driving this, along with a few other ones. And so it kind of hints at the fact that there's some kind of constraints which are responsible for seeing this repeated evolution. All sorts of other more polygenic examples have been found. We were just working on this project on local adaptation in conifers, where we looked at adaptation to climate, and found 47 genes with strong signatures of adaptation in these two very divergent conifer species. So we can see highly polygenic responses also being highly convergent. And again, this question of is the convergence that we're seeing caused by constraint, and what do we want to, what can we conclude about this, and how can we learn about the factors that are shaping this repeated evolution? Some of the factors that people have talked about before that we think about why this happens over and over again, things like mutation rate, where if some genes are more mutable, if you can break a gene more easily then you can fix it, so if you have loss of function mutations, that can be very easy. Things like the number of loci, or the number of mutations that can yield a similar phenotype, so how genetically flexible a trait is. If there's lots of different ways to yield the same thing, then that's clearly a less constrained scenario. All sorts of things like pleiotropic costs and epistasis can then shape also whether or not a gene, if it has a lot of negative side effects, when you mutate a gene, it's not likely to contribute to adaptation, so that can really drive the repeatability. And then also considerations like effect size in the context of either migration selection balance or drift selection balance can also be a constraint on adaptation, and a constraint on kind of why the same form evolves over and over again. And so I kind of classified these into broadly genetic constraints and selective constraints, only because I think that classifying this kind of helps us understand why we really care about this, right? And I think that genetic constraints you can kind of think of as saying that some forms are more possible than other forms. They're coming up more easily, more readily. Whereas selective constraints we can think about as being saying many forms are possible, but few of them are fit enough to actually make it through that filter of natural selection. And I think these are important things to think about because they represent very different worldviews of how evolution is working. And I think it's really fascinating to understand, is it that evolution is able to generate lots and lots and lots of things, but only a few of them are fit enough, or is it that our developmental genetic program is actually kind of canalized and stuck going in a certain direction? So I'd really like to think about why constraints happen, and if there's some kind of broad organizing principles we can make about this. And to do this, what I'm going to talk about here is thinking about ways we can actually formalize and quantify hypotheses about constraint. So I'll set up a little toy model to think about this. Two species with genes that are locally adapted, either in dark or non-locally adapted in light. And thinking about how to test hypotheses here, say we have 50 genes in total, and 25 that are locally adapted in one species, 10 in the other, and 9 that are shared between both. This matches with the hypergeometric distribution. So if we kind of assume that there's no constraints under some null hypothesis, every gene has an equal probability of contributing to adaptation, then making random draws by the scenario matches the hypergeometric, which is really nice because it has properties like an expected mean and standard deviation that we can use in deriving some kind of effect size metrics and testing hypotheses about this. So if we look at the hypergeometric distribution, what it predicts in this particular scenario, this is the number of genes that are overlapping in a given case, and then the probability of that occurring on the y-axis. And we can see here our observation is much more overlapping than expected under the hypergeometric distribution. And we can calculate p-values by asking, what are the probability, the sum of all the probabilities that are as extreme or more extreme than our observation. That's nice. But what I think is more interesting is quantifying the effect size, because that gives us a metric for saying, how convergent is this? And so what I propose here is to use a z-score, where you just take the observation of number of overlaps minus the mean and divided by the standard deviation is one way to kind of quantify, how many more standard deviations above the average expectation are we in terms of how much constraint we see in this system. Just to kind of illustrate what happens if we kind of dial up the number of genes in this scenario, right, where there's more potential genes we're drawing from, then the chance of seeing 9 genes overlapping becomes less likely, and so that probability distribution gets shifted towards the left in this case. And so then our observation has a lower p-value, it's less predicted, and we also have a higher z-score, right? We're further away from that mean expectation in terms of standard deviation units, and so our measure of constraint, this z-score, gets higher. What's nice about this approach is that it is able to differentiate between these two scenarios. So cases where we're drawing from a different number of genes, where other metrics that have been used in the literature to kind of quantify convergence and repeatability, the Jacquard index or proportional similarity, to name a few, don't differentiate between these two scenarios, right? They both measure a similar amount of repeatability, 0.35, because the actual genes that are adapted are the same, but they're not accounting for the probability and the fundamental kind of how unlikely this is. And so I think it's nice to bring that in, because it's explicitly more about a hypothesis test. And so I think it's really important in this context to think about what the meaning of G is, right? G is the number of genes we're drawing from, and setting different values of G can allow us to test different kinds of null hypotheses. So the first and most obvious one is just assuming that there's no constraints, that we set G equal to the number of genes in the genome, and we just say, any gene could potentially contribute to adaptation, and let's see how much more constrained we are than the expectation of just making random draws. That's clearly a bit of a weird biological null hypothesis, but I think it's a really nice one to start with, because it gives a very simple way of formalizing constraint. We can also then kind of modify this. For example, if we know that a given gene or a given trait tends to be driven by, say, 50 loci, then we can set G to be much smaller and say, well, this particular trait, we know it only mutations in these subset of genes will actually give rise to variation, and so we can then test, refine our null hypothesis and test that. We can also do other models where we could fit, pull different weights for different genes if we thought they were more or less constrained for selective reasons, which I won't go into. But basically, the point here is that observing significantly more overlap than expected by any given null model is just a way of saying there's unexplained residual constraints that are unaccounted for in our null model, and that's something interesting that we learned from this. So I think that's what I'm trying to get at here, is how this could be useful. I won't go into it, but we've done some work generalizing this to multi-species versions. I should say multi-species, not multi-locus. But this is using the chi-square distribution, and it's broadly collinear. So if I ever get a paper together on this, you can read about it there. To give some intuition about how this metric is working, I ran some individual-based simulations using NEMO. So this is running 20 replicate populations and then doing that 20 times, and then taking the average, how much constraint is observed in each of those scenarios. On the x-axis here is the number of coding loci that are responsible for driving a trait out of 100. So sometimes you've got 10 coding loci and 90 neutral loci, or 100 coding loci. And you can see that as you go from to more and more loci, as we go right on that case, we get less and less constraint. That's just because as you have 100 loci, there's many, many different ways of drawing your locally adapted type. This is under migration selection, two-patch model. And so as we get towards the right, the constraint decreases. And so it's just showing that our metric here, this ZX metric of how much constraint is in the system, kind of behaves as we might intuitively expect when we ramp up or down the number of loci. Same kind of deal for selective constraints. In this case, it's here simulating, again, migration selection balance with a low migration rate, and changing the gamma distribution of effect sizes that are at particular loci. And so if we're all the way over on the right, in this case here, that's basically saying that every single effect size is the same, whereas if we're over on the left here, here there's going to be one very large effect locus and lots of small effect loci. The same locus in every single replicate of the simulation is a large locus, and what you can see is that there's constraints driven by this selective differences, right? So the ones on the left are much more constrained than the ones on the right, and that's kind of just behaving according to our intuition. So that's nice. We kind of started thinking about this in the context of this lodgepole pine and white spruce study, just to give an example of how you can use this in an empirical context, maybe. In this case, for one particular environmental variable, we found 71 locally adapted genes in pine and 139 in spruce, and 5 that were in both. And what you can do is then, plug those into this formulation, assuming we studied a total of almost 12,000 genes. This is, if we were doing a random draw against that null hypothesis that every gene could have contributed to this, it would result in a z-score of 0.46. But we can also flip that around and say, well, how big would the pool of loci have to be to have gotten no constraint, right? To have gotten a z-score equal to 0, and that's that red line there, so we can kind of look down that and say, well, if there were 2,000 loci, that would give us random draws of getting, 5 loci overlapping on average. So, I mean, I don't think that's truth. This is just an example of how things can work. And the reason I don't think it's truth is because we don't know how many false positives were in our particular study. So, if 50% of the loci were false positives, then those numbers change a whole lot. And so this is an ongoing empirical problem, is actually how you get good confidence on what genes are locally adapted and what aren't. But, that's a talk for another day. So I think what I'm trying to say here is I think that this approach to quantifying constraints using the z-score approach gives us this robust method to quantify how much repeatability there is in a explicitly hypothesis-driven way. And it's robust to changes across the number of mutations involved, the number of genes involved, and the number of lineages that you're contrasting. So it's able to kind of generalize to any number of contrasts you want to make across your phylogeny. As I said, it's explicitly suitable for hypothesis testing because it's firmly grounded in probability. And what I hope is that it opens up new ways to explore how evolution is flexible or inflexible, and why diversity of forms at the genomic level evolves or doesn't. So there was this paper that came out this week that probably a lot of people read and were interested in and thinking about it, and it made me think, oh, genome scans are dead, everything is actually contributing to variation, but no. I mean, I think that it's a more nuanced perspective here probably, and we do find convergence, we do find repeatability and signatures of things that are interesting, and so I think, I think there's a lot of scope for actually using these kind of approaches, and I think failure to find convergence is just as interesting as finding convergence, because that implies that there's lots of different ways to do things, or that we've just not done our stats well. So the kind of thing I'd like to think about doing with this work is to kind of compare the ZX value among things like trait types, looking at, say, morphological, physiological, behavioral traits. Does it differ? Do the kinds of constraints that shape these differ? Trait complexity, levels of organization as you kind of integrate across more tissue types or organ systems, that kind of thing. Things with different NE, we might expect the efficiency of selection might really drive the kinds of constraints that evolve in different systems. So if there's comparing multicellular eukaryotes to single-celled things that have enormous population sizes, we might see different kinds of constraints operating. And things like new versus old lineages, if there's things that have been long statically, not radiating versus new bursts of evolution, that might be also a kind of interesting axis, just to name a few. So that's as far as I got with this. I'd like to really acknowledge Les Harris. Elise and I were sitting down having a beer talking about this weird probability model we were trying to formulate, and Les was like, just use the chi-square, and it was great. So, thank you for listening. I have to take questions.